Hello students, Professor Hadke here. Today we discuss about our unit 5 of biotechnology. In unit 5 we have to discuss about the production of different human healthcare product by using recombinant DNA technology. Now in that first point here we have studied that human insulin. Now you know that this human insulin or the insulin is nothing but the protein which is to be produces in a pancreas. In pancreas, that isolate of Langerhans are present. In isolate of Langerhans, that beta cells are present, and the beta cells are responsible for the production of that human insulin. Now that human insulin which is to be produces, now first that insulin made up of 109 amino acids, and then when that insulin is called as a proinsulin, then this insulin after production then reaches the blood. Then when this insulin reaches the blood, now some amino acids are deleted, and then finally that 51 amino acid protein, that is nothing but the insulin, reaches that blood, that is simply normal insulin. Now what is the role of insulin? That you know that the first that role of insulin, that this protein normalize the blood glucose level. Now how this insulin normalize the blood glucose level? Now that the glucose when increases in a blood, that the extra glucose remove from that blood and then transfer in a cell by that insulin, and now in a cell that insulin or that glucose is used for the production of energy, that by the process of glycolysis, that you know, and then that energy are produces in the form of ATP and utilizes that cell for its activity. Now second thing, again that remaining glucose then convert into the glycogen, and then that glycogens then stored in adipose tissues. Whenever there is a that lacking of that glucose, then the glycogen convert into the glucose and the glucose level is maintained. Now these are the that role of that insulin. Now in some person that insulin is unable to, normal insulin is unable to produces, that normal insulin made up of 51 amino acids, that normal insulin unable to produces, okay, and then that condition we say that is a diabetes, okay, you know that. And then the diabetic patients, now in a diabetic patient the blood glucose level are increases than normal, okay, and now to treat that diabetic patients, now from outside that insulin are injected in a blood in a body and the glucose level is maintained, okay. Then this is about some introduction about the insulin, but here our heading is the production of human insulin by using recombinant DNA technology. Now here, as I said, that insulin which is to be injected in a diabetic patient to normalize the blood glucose level, then that insulin are produces in a industry by the process of recombinant DNA technology. Now here that first I said that is a pre-pro insulin. This pre-pro insulin which is initially made up of 109 amino acid, when this insulin transfer through cell membrane of beta cells, the first 24 amino acids are deleted, that the amino acid remain 85 amino acids. It is called as a proinsulin. Again that proinsulin when through that tissue fluid when reaches the blood, now that again that 35 amino acids are deleted with the help of endopeptidase enzyme, and finally 51 amino acid insulin, this is a normal insulin which reaches the blood, okay, and perform its normal role, that is a normalization of the blood glucose level. Now as I said that this insulin in industry produces by the process of recombinant DNA technology. Now the student, before that again one point is that the human insulin made up of two polypeptide chain. Now, as I said, the human insulin is nothing but the protein. Normal protein made up of 51 amino acid. Okay. Now this human insulin made up of two polypeptide chain A, which made up of 21 amino acid, and chain B, which is made up of 30 amino acids. This chain A and chain B join together by using disulfide bond. Okay, this is the complete structure of human insulin here. That we just keep in your mind that this insulin, normal insulin, okay, or the insulin which is to be present or produces by pancreas, okay, this insulin made up of 51 amino acid. The structure of insulin, this is made up of two polypeptide chain A and then chain B. These two chains are joined together by using disulfide bond and form the normal insulin. That's why taking this structure, by considering this structure, now in industry, this insulin is produced by using recombinant DNA technology. This is our main point. Now for that, you know that this insulin are produced in a bacterial cell. That bacterial cell, that is the E. coli cell. In E. coli cell, this insulin is produced. Okay. Now again, you know that insulin is nothing but the protein. Hence, for the production of that insulin, DNA is responsible. Okay. What happened here? That the structure of insulin chain A and then chain B, these two polypeptide chains are separately produced in a bacterial cell. Now for that, the first gene is isolated or that synthetically prepared. That gene A, this gene A responsible for the production of polypeptide chain A, which is made up of 20 amino acids, and the second, that gene which is to be used, that gene B, responsible for the production of polypeptide chain B. These two genes are separately joined to the vector. Now here the vector is used, that is PBR322, that you know the name of that vector, PBR322. P stand for plasmid, B stand for Bolivar, and then Rodriguez. Okay, this is the name of that vector. Now the structure of the vector, you know that is it having the, now here the normal structure. In that, multiple cloning sites are present where the recognition sequence of EcoR1 is present. Second point here, second point is that is ampicillin resistance gene which is to be present. Ampicillin resistance gene, ampicillin resistance gene which is to be present. You know this, the role of that ampicillin resistance gene, that is selection of transform cell. And then another point which is to be present, that is origin of replication. Origin of replication. Now this origin of replication is required for the replication of that vector or the plasmid in a bacterial cell. The same structure is here also. Now that this gene are separately integrated or attached with this vector. Gene A attaches to this vector, gene B separately attaches to the second PBR322 vector. And now you know that after attaching this vector, now the vector which is to be prepared, now it is called as a recombinant PBR322 vector. You know that recombinant PBR322 vector, that the vector to which that gene which is to be inserted in a bacterial cell, that first that gene A are combined with the vector, then gene B combined separately with the vector, and now here the two recombinant plasmid is produced. Now this recombinant plasmid then transfer in a bacterial cell. Here bacterial cell, that is the E. coli cell. Now again you know the transformation process, that in chapter or unit 3rd we studied about the transformation process. Okay, how this recombinant vector transfer into the bacterial cell. Okay, that is transformation. Then this recombinant vector transfer in a bacterial cell. First recombinant vector which having the gene A, second recombinant vector which having the gene B separately transfer into the separate bacterial cell. And then after that, again you know that selection process, we require the transform cells. The transform cells means the cell which having the recombinant vector, that cell is called as a transform cell. The transform cells are selected by screening method. Screening method, you know that. Now here the ampicillin resistance gene is present, that's why these bacterial cells are grow on a antibiotic containing media. You know that transform cells only grow on a antibiotic containing media. Non-transform cell does not grow because in a transform cells recombinant vector is present. On a recombinant vector, that ampicillin resistance gene is present. That gene resists the activity of that antibiotic. Okay, this is about the transform cells. What about non-transform cells? Non-transform cells does not grow on a antibiotic containing media. Why? Because in a non-transform cells this recombinant vector is not present, means that ampicillin resistance gene is not present. Okay, and finally we require the transform cell, and then these transform cells are that then grow in a fermenter. Okay, because here our aim is to produce the human insulin protein in a large quantity. That's why here this bacterial cell, here we draw the single bacteria, but there is a number of bacteria present, number of transform cells are present. These transform cells then grow in a fermenter. You know the fermenter, in that media is present. On that media these transform cells are inoculated, and now that this bacteria take a nutrient from that media and start its multiplication, the number of bacterial cells are produces. Now when the bacteria multiply, divide, simultaneously this recombinant vector also divide. The gene which is to be a present on a recombinant vector also multiplied, expresses the expression of that gene A, and then gene B is nothing but the production of protein, that polypeptide chain A and then polypeptide chain B. Then now one thing is that one gene is present in a vector, is a beta gal gene. These beta gal gene are present in combination with the gene A in a first vector and gene B with the second vector. That's why simultaneously this beta gal gene also expresses, and finally now after that there is a harvesting. Harvesting means that isolation of this bacteria from the fermenter, and now the purify the protein, that insulin. In a first bacterial cell, that polypeptide chain is produces, that protein is produces because in having the gene A. Gene A produces the polypeptide chain A, which is made up of how many amino acid? 21 amino acid. In a second bacteria, in a second transform cells, gene A B is present. This gene B produces the polypeptide chain B. Now here in addition with this chain gene A and then gene B, beta gal gene is present. That's why this beta gal gene also expresses and produces the protein beta galactosidase. But here there is a combined protein is produces, that chain A, polypeptide chain A plus beta galactosidase, polypeptide chain B plus beta galactosidase. But here we do not require the beta galactosidase protein. That's why these beta galactosidase proteins are separated, or that remove from that chain A and then chain B. Now remove beta galactosidase from chain A and then chain B by using cyanogen bromide. This cyanogen bromide deleted that beta galactosidase from chain A and then chain B, and finally we have here polypeptide chain A and then polypeptide chain B. Okay, now here two polypeptide chain are produces. This polypeptide chain A made up of 21 amino acid, and polypeptide chain B made up of 31 amino acid. Okay. Now next step is that these two chains are joined, because the structure of insulin is chain A combined with the chain B by joining the disulfide bond. Okay, that's why here chain A, which is composed of 21 amino acid, which is to be produces here, chain B composed of 31 amino acid, which is to be a produces here, combined together by forming disulfide bond by using sodium sulfonate. Now here sodium sulfonate are used, and this sodium sulfonate combine this chain A and